Assalamu alaikum, good morning, good afternoon, and good evening to the people across the globe who have joined us for today's webinar. I'm Dr. Anna Musa, Associate Lecturer in Department of Plant Pathology, the Islamic University of Bahawalpur. I'll be your moderator for today's session, and the session will be co-moderated by Dr. Taimur Shakil, Assistant Professor, Department of Plant Pathology. On behalf of Department of Plant Pathology, the Islamic University of Bahawalpur, and Pakistan Phytopathological Society, I feel pleasure to welcome you all for joining us today. In continuation of our webinar series, today's session has been organized to discuss stealth pathogens, the sooty blotch, and fly-speck fungal complex. I feel honored to introduce our distinguished speaker and guest for today's session. Have been kind enough to adjust their professional commitments and busy schedules for sparing time for this session. We all are thankful to worthy Vice Chancellor, Professor Dr. Athar Mehboob, Dr. Muhammad Iqbal, Dean Faculty of Agriculture and Environment, Dr. Moazzam Jameel, Registrar, the Islamic University of Bahawalpur, Professor Dr. Tariq Mukhtar, President, Pakistan Phytopathological Society, and the organizer of today's session, Dr. Muhammad Naveed Aslam, Chairman, Department of Plant Pathology. And very special thanks to our honorable speaker, Dr. Mark Lawrence Gleason. Dr. Gleason has been very kind for giving us time for this webinar. Dr. Gleason is serving as President of American Phytopathological Society and Professor of Plant Pathology in Iowa State University of Science and Technology. Dr. Gleason has given us the opportunity to several postgraduate students from Pakistan who have worked in his lab as visiting researchers. So he has been sharing a long-term association with Pakistan. I'm glad that today he will be sharing his valuable work with us. So now I would like to formally begin this session, and I will request Dr. Muhammad Naveed Aslam to share his thoughts with the participants. Over to Dr. Muhammad Naveed Aslam.
I'm sorry. Thank you, Dr. Anna, and Assalamu alaikum, everyone. Good morning, good evening, good afternoon, and good evening, and greetings from Pakistan. I hope most of you are having a comfortable fasting today. On behalf of Department of Plant Pathology, the Islamic University of Bahawalpur, I welcome you all to this second session of the series of international scientific webinars arranged with the purpose of capacity building of plant scientists and students of plant pathology. I warmly welcome the Vice Chancellor. I warmly welcome the worthy Vice Chancellor, the Islamic University of Bahawalpur, Engineer Professor Dr. Athar Mehboob, for sparing precious time for us. Sir, this is an honor for us and encouragement as well that you have joined us our session. I would also warmly welcome to our guest speaker, Professor Dr. Mark Gleason, who is the President of American Phytopathological Society and serving Iowa State University of Science and Technology, Ames, Iowa, USA. We are pleased to have the presence of Professor Dr. Muhammad Iqbal, the Dean Faculty of Agriculture and Environment, and the Registrar, the Islamic University of Bahawalpur, Dr. Moazzam Jameel. We are also honored to have the presence of President of Pakistan Phytopathological Society, Professor Dr. Tariq Mukhtar, who is serving the Arid Agriculture University of Rawalpindi. I would appreciate the participation of today's session for different universities and research institutes of several countries, including Pakistan, USA, Australia, China, Saudi Arabia, Argentina, Costa Rica, South Africa, India, and Nigeria, and from other countries. We have received an overwhelming response from our participants for the previous session and this one too, as more than 1,000 scientists responded for this webinar. The Islamic University of Bahawalpur holds a key position in the southern Punjab of Pakistan and therefore has always focused on the capacity building and grooming of the students by promoting the research culture among the scientists while they are at the developing stage. Ladies and gentlemen, I believe that by participating in this webinar, we are in the right place and right time today. Together, let us accelerate the exchange of ideas and scaling up of good practices. Before we get started, I would like to go over a few items so you know how to participate in today's event. We have shared a live Facebook link in WhatsApp group. If you went disconnected at any stage, you can join through that Facebook link. You will have the opportunity to submit text questions to today's presenter by typing your questions into the question pane of the control panel. You may send in your question at any stage, any time, and during the presentation, we will collect these and address them during the question answer session at the end of today's presentation. In the end, I will once again welcome the worthy Vice Chancellor, Professor Dr. Athar Mehboob, Professor Dr. Mark Gleason, the Dean Faculty of Agriculture and Environment, the Registrar of IUB, and Professor Dr. Tariq Mukhtar, the President of Phytopathological Society. I wish you a very successful webinar. Now, I request worthy Vice Chancellor, Engineer Professor Dr. Athar Mehboob, to say a few words as we begin with the events. Sir. Sir, ka mute mic on kare.
Okay, thank you very much, Dr. Naveed Malghani. I hope I am clearly audible.
Yes, yes, sir.
Okay, Bismillahir Rahmanir Rahim. Welcome to everyone and a very good evening to everyone in the Eastern Hemisphere and a very good morning to everyone in the Western Hemisphere. Remarks to appreciate the efforts of Dr. Naveed Malghani, the chairman of the Department of Plant Pathology at the Islamic University of Bahawalpur. The success of the event can already be judged not only by the keynote speaker and the other worthy moderators and the panelists and the organizers, but also by the huge response of more than 1,000 participants seminar. And I would not come for a long time in between the appreciation for the Department of Plant Pathology, for Dr. Naveed Malghani, the honorable guest speaker, Professor Dr. Mark Gleason of the Iowa State University. Thank you very much, sir, for your valuable contribution by being the keynote speaker today. And I would also like to thank Dr. Tariq Mukhtar, the president of the Pakistan Phytopathological Society, the dean of the Faculty of Agriculture, Dr. Iqbal at the Islamic University of Bahawalpur, and the former principal and currently the registrar, Professor Dr. Mozam Jamil. So I won't hold you any longer from proceeding further, and I wish that all the participants have a very valuable session today. Thank you very much.
Thank you very much, sir, for your valuable time. Now, I will request Dr. Annam Musa to proceed further.
Okay. I would like to request Dr. Mark Lawrence Gleason to please, he can please start his talk.
Well, thank you, Dr. Musa. Can everybody hear me okay?
Yes, we
Yes.
can hear you.
Very good. I'm honored by this invitation from the Islamia University of Bahawalpur, and I'm going to talk about a subject that I have been working on for a number of years, and I'm going to switch now to show you my PowerPoint. Does that look okay, Dr. Musa?
Yes, it looks okay. Perfect. Fine.
Very good. Again, thank you to the organizers. I feel very privileged for this invitation. So my topic today is stealth pathogens, the sooty blotch and fly speck fungal complex. What does stealth mean? Well, it means it by the dictionary, slow, deliberate, and secret. These are fungi that are slow growing, often overlooked, misunderstood, and not very much studied. I'll cover parts of 6 different subjects concerning sooty blotch and fly speck: taxonomy, biogeography, management, phenology, evolutionary phylogeny, and adaptation to what's a rather unique niche. The sooty blotch and fly speck group makes up a common fungal disease of apple fruit in many places around the world, including Eastern North America, where I live. By the way, this group also occurs, and some of you may have seen it, in Pakistan, I believe in Rawalakot area, where apples have been grown. And so this is kind of a worldwide group of fungi. These fungi are fairly distinctive in that they colonize the surface of many plants, not only fruit, as you see here on the left, but also stems and leaves and other waxy coated plant surfaces. In Eastern North America, this group of fungi is economically important. So a lot of fungicide sprays are applied every year to control this group during the fruit development period, because generally it's difficult to sell these fruit as fresh fruit if they have spots and blemishes on them. They have to look really good for the market. The story of the study of sooty blotch and fly speck goes back nearly 200 years. The first publication on sooty blotch and fly speck was published in the United States in 1832 by a person who is now considered the father of mycology in the United States. At this time, mycology was entirely morphology based. So mycologists, and there were a few of them at the time, mycologists relied on characterization of the visual impression of colonies and also the micromorphology based on what they could see through the microscope. For a great majority of the time that this group of fungi has been studied, they have given a lot of frustration to mycologists. They're somewhat difficult to study for a number of different reasons. One problem is that they grow very slowly in culture. And the picture I'm showing you on the lower left is a colony that has been growing for 3 months on an agar plate, on a PDA, potato dextrose agar, and it's only 1 centimeter across. And you can imagine if these fungi are so slow growing that it's rather difficult to isolate them, because many other fungi, which also live on the surface of apples, are faster growing and will overgrow them. So contamination during the isolation process makes one sort of challenge. Here's another challenge that mycologists gradually realized, pardon me, and that is that there are many cryptic species within the sooty blotch and fly speck complex. What exactly are cryptic species? Well, cryptic species are species that may function differently, but to the naked eye they look very similar. They look alike. So how to sort out this mass of different fungi was a huge challenge. So here we have to cross this bridge of taxonomy in order to be able to understand how these fungi function and how to better control them when growing apples or other fruit that they colonize. And you can, in this photograph, you can see, or this set of photographs, you can see that there are many different colony types. These are just close-up pictures of the apple peel, and you're looking at different, what's called mycelial types on the surface of the apple. So we have a lot of different mycelial types, and the real question has always been, which species is which? A great deal of effort went into taxonomy, and also a great deal of frustration went into taxonomy for many, many decades. We were fortunate in that we began our study of sooty blotch and fly speck here in Iowa, USA, about the time that the tools of molecular genetics came into wide use around the world. These were enormously helpful to us in understanding some of the secrets of this group. We started by surveying a number of apple orchards in the US. We did 2 surveys at 2 different times. And this is how we did those surveys. We would find apples that had colonies of sooty blotch and fly speck fungi. And we would sample about 40 apples per orchard that displayed these colonies. And we would count the number of colonies of each mycelial type. And here you see the mycelial type. We characterized about 8 of these. The first step we decided to do was to use acidified water agar as the initial isolation phase, because we knew these are very slow growing and we did not want to encourage the growth of faster growing fungi. So we began isolation with acidified water agar. As part of this process, we also preserved the peel of the apple. We would dry that peel and press it and make essentially a permanent record of that colony from which we had isolated, so we could always refer back to that. And once we got some decent looking colonies on acidified water agar, we would go to potato dextrose agar, and then use molecular genetic techniques to amplify the ITS and LSU regions of ribosomal DNA and sequence. And so we would sequence these characteristic regions. ITS, as you know, is considered a universal barcode for fungi, and that was a good starting point for us. And at the same time, we use the traditional morphologically based methods of mycology in order to characterize the spore morphology, conidiophore morphology, et cetera, under the microscope. This was extremely necessary in order to be able to name some of these new species that we found. This effort combining genetic analysis or molecular genetics with morphological characterization really changed the picture in terms of our understanding of sooty blotch and fly speck diversity. For a long time, from about 1920 to 1997, sooty blotch and fly speck was considered 2 diseases, each disease caused by one species. So sooty blotch, which is that colony type that you see in the red circle in the picture, that's abbreviated SB. was thought to be caused by one fungus called Gliades pomigina. And fly speck, which is that group of dots that you see on the lower right of the picture, was thought to be caused by one fungus by the name of Schizotherium pomi. So this was the dogma in the world of sooty blotch and fly speck for nearly 80 years. But then things began to change with the use of that new tool, the molecular genetics. This is when it began to change. 1997, work at North Carolina State University showed that there were 4 species in the sooty blotch and fly speck complex, not 2. 2 new species were described. They were still using the traditional mycological methods, but then when we combined molecular genetics, this is the picture that emerged. In 2021, we recognized more than 100 species in many genera and several families within this complex. So this is not certainly unique in the world of mycology. We're kind of living, I think, in a golden age of studying mycology because we have these tools at our disposal, and many things that we thought were perhaps one species or a simple group turned out to be more complex than we realized. And that's certainly true for the sooty blotch and fly speck complex. And this number of species in the complex continues to rise as it's studied around the world. This led us to realize that we have to think about sooty blotch and fly speck fungi in a new way, a different way, a new paradigm, if you will. This isn't really 2 diseases called sooty blotch and fly speck, but rather, instead, this disease is a multi-species complex. It's true that you can look at certain colony types and say, oh, those look like fly specks. And you can look at another type and say that that looks like a sooty colony or sooty blotch. But in fact, in any orchard in the world, you have a mix of colony types that are those end types, but also many intermediate. So we have, in fact, a multi-species complex. In any given orchard, we may have anywhere from 2 of these species in the complex to as many as 13 species in the same orchard. Number of papers that were published along the way, bringing home these findings. The most recent was an annual review in 2019. Okay, well, it's a good start, I guess, to characterize the taxonomy of the group, and that's how we got into this work in some depth. But then, what do you do with that knowledge? And we had, by this time, accumulated a lot of isolates, and a number of other people around the world were working on sooty blotch and fly speck as well. And some of these people we brought in to work on sooty blotch through various partnerships, and they were very productive. So you can see apples and similar fruit are grown in many parts of the world. And these places where the small apples are located are places that we've had collaborations with. Unfortunately, we've never worked with anyone in Pakistan, although I have tried in the past to do that. We would love to be able to have a collaboration with Pakistan on characterizing the diversity of sooty blotch and fly speck fungi in your country. So thinking about this topic of biogeography, which I think of as which species live where. We first turned to our surveys from the US. We surveyed a total of 39 orchards in many places within the eastern US. And what we found was that certain sooty blotch and fly speck species, and you can see 3 of them listed here, had a distribution in space that we characterized as cosmopolitan, meaning that basically almost anywhere we looked, almost any apple orchard we looked, we were able to find these fungi. Schizotherium pomi, Pseudocercospirella, Peltaster fructicula, basically were distributed everywhere, and often these same species reoccurred in many locations around the world. But then in contrast, there are other species that could be characterized as regional in their distribution. They were not located everywhere that we looked. Some of these, like Geostrumia, Polystigmatus, Phyalophora sessilis, and Stomia peltis, SP 5.1, these species were restricted in their distribution or regional. Some occurred only in the Southeast US, some only in the East, some not in the upper Midwest. This was really the first work that anybody had done to try to characterize which species lived where. And you could not have done this work without the molecular genetics knowledge that enabled us to separate these species from a mass of similar looking fungi into distinct species. So this is just the basic, I think, what you might call the beginning work in the characterization of biogeography. I work with apple growers in Iowa. I have for many years. And I worked many years in extension as well, as we call the outreach to growers. And this is how I got into studying sooty blotch and fly speck work in the first place, because growers spend a lot of money and time spraying fungicides in the summer as the fruit are developing in order to control sooty blotch and fly speck. So one question that we asked is, can we make that control more cost effective? Can we be more efficient in that control? What you're seeing here is an air blast sprayer, very typical of what's used in apple orchards. The first work that was done to try to make fungicide spraying more efficient was done at North Carolina State University by Dr. Turner Sutton in the 1990s. He developed the first, what we call a warning system for sooty blotch and fly speck. It's a rather simple warning system, which that tends to be the best kind of warning system that's simple to use. And about the time of first cover, which is maybe 10 days after the flower petals fall off the apple trees, you withhold the next spray until 175 hours of leaf wetness have accumulated since that first cover spray. And then you begin a normal program of sprays thereafter. In some years, in a dry year, that might save 2 or 3 sprays, maybe 4 sprays. So on average, it would save about 2 or 2.5 fungicide sprays per year without compromising the control of sooty blotch and fly speck. So this was a way for growers to save money and save time and become more efficient in their control of sooty blotch and fly speck. Okay, so this story is a North Carolina story, but North Carolina is a long way away from where we live in the upper Midwest. And so there's always a question when you develop a warning system in one place. Can you move it like a suitcase from one place to another? How portable is that warning system? And we tried this. We tried to take the North Carolina state warning system and move it about 1,200, 1,400 kilometers away to the upper Midwest US. And we found out that it works sometimes, but when we got into orchards of commercial orchards of actual growers rather than university orchards, we found that it did not work consistently. And that's a very important criterion for a warning system. It has to work very consistently. And we published that work some years ago. So that told us that if we're going to use a warning system, a weather-based warning system in the upper Midwest, we would have to change it. We would have to do something different. So we basically started over. We monitored a lot of weather conditions in many different orchards over several years in Iowa and Wisconsin. And we monitored temperature, relative humidity, rainfall, and LWD, which is leaf wetness duration. Leaf wetness duration is the number of hours per day when the foliage is wet. So these 4 weather variables we monitored hourly, every hour, over 19 orchard years in the upper Midwest. And what did we come up with? Oh, I should mention a graduate student on the project, her job was to go out to the orchards and find out when the first colonies appeared on the apple. So she went out every week and watched and watched and watched. And towards the end of the season, she began to see little colonies starting to appear. And this is where a grower starts to lose money because the apples are appearing to be blemished. So this was another marker that we needed. We know that a commercial apple grower would need to apply a spray before these colonies appeared. Let me explain what we did here. It's not complicated. It has a fancy name. It's called receiver operating characteristic curve analysis, or ROC analysis. This came into use in the plant pathology world about 20 years ago, 15 or 20 years ago. And it's just a way to evaluate how accurate your warning system is. So we tried to characterize, we developed a number of different warning systems based on our monitoring of the weather. And that the analysis in this ROC is very simple. It's based on what's called a contingency table. This is an analytical method that did not start in the world of plant pathology. It started in the world of human medicine, in the world of cancer diagnosis. And there certain predictions are made. And then the question is, how accurate were the predictions? And that is exactly our question. If we make a prediction about the timing of appearance of sooty blotch and fly speck, how accurate are we? And there's really 4 different outcomes in this contingency table. The first outcome is what's called the true positive. That means I predicted, my model predicted, that sooty blotch and fly speck colonies would begin to appear on apples at a certain date. And the true positive outcomes meant that, yes, in fact, that happened, that sooty blotch colonies appeared when I predicted that they would. So that's called a true positive. Another outcome is the true negative. Also, you could call a desirable outcome. That means that I predicted that the sooty blotch and fly speck fungi would not appear at a certain time. And yes, they did not appear at that time. So those are the true negatives. Those are what you would call the accurate outcomes. But what really determines whether the model is useful is not only that, but the errors. And here we get into 2 kinds of errors. One kind of error is false positive, which means that the model, based on weather information, told us that sooty blotch and fly speck colonies were going to appear at a certain date, but lo and behold, they did not appear. This is a mistake of the model. That's called a false positive. The other kind of error is false negative, and this is by far the worst kind of error, because here the model said, no, sooty blotch was not going to appear in a certain date, but it did appear. The false negative is the worst kind of outcome for growers, because this is where they lose money. This is where they have spots on their apples. So a good model, a reliable model, must absolutely have the highest percentage of true positives and true negatives, but especially have a very, very, very low rate of false negatives. So this was kind of our principle that we used to develop a new model for the upper Midwest for sooty blotch and fly speck. And this is what we proposed based on just essentially crunching the numbers. We found, according to this analysis, that for our climate in the Midwest, relative humidity rather than leaf wetness was a better predictor of sooty blotch and fly speck activity. And our model looked a little bit different than the one from North Carolina. This warning system indicated that it was a gap of 192 hours of relative humidity greater than 97%. You remember the previous model was based on leaf wetness duration. This one is based on high relative humidity, number of hours in which relative humidity exceeded that threshold. Well, that was a starting point for a new model. And we began to evaluate that in the field, because it's one thing to play games on a computer, but it's another thing to see what works in the field. And this led us to actually modify this system. We did field trials over this period, 6 different years. And over that period, using the warning system saved about 2.5 sprays per year. But we also found that 97% relative humidity was just too high for what's called an action threshold. We needed a threshold that was lower than that. So we went down to 90% relative humidity. It turns out that humidity sensors are not very accurate at very, very high RH. So we found a lot more accuracy from sensor to sensor down at 90%. That led us to this model, which is the one we published a few years ago and we're using with growers today, where we have 385 hours of relative humidity greater than 90%. That's our current model. Seems to be the most reliable model for conditions in the upper Midwest. So this is a story of the adaptation of a warning system from one region in the Southeast US with quite a different climate to another climate in the upper Midwest US. This is fairly typical of the need to change or at least examine a warning system before it's moved from one place to a faraway place. Well, that's all I want to say about management. The next topic I want to talk about is phenology. And phenology is just the pattern of seasonal occurrence of various phenomena in nature. And here we were interested in when apples became inoculated by sooty blotch and fly speck spores. These spores fly through the air, land on the apple at different times. And we would like to know, now that we could identify which species is which, we'd like to know whether there are differences among the sooty blotch and fly speck species in terms of when they arrived on the apple. And I'll explain how we did this. This was the PhD, part of the PhD thesis of a student from Malaysia, Zara Ismail. And Zara did this experiment in the field in our university orchard. Let me explain what's going on here. It looks complicated, but basically it's not. So if we look on this chart from left to right, we're looking at different 2-week periods during the growing season. Period 1 is when the fruit are very small. Period 7, you're just about to harvest. And the different treatments that Zara imposed in her orchard were basically to expose different groups of apples to possible spores landing on them for 2-week windows during that season. So she had 7 different 2-week windows when those apples were exposed. The rest of the time, the apples were enclosed in these fruit bags that you see. So we would wrap the fruit bag around the individual fruit so that spores could not land on it. But during a 2-week period, the chosen 2-week period, those apples were exposed. Then at the end of the season, Zara looked at all the apples and found out which colonies had formed. We did PCR to find out, to confirm which species were involved, and we came up with some patterns. What this graph shows, we have 1, 2, 3, about eight different taxa, 8 different species of sooty blotch and fly speck here. What this graph shows is that there are differences among species of sooty blotch and fly speck as to when those spores land on the fruit. You'll notice the x-axis is a variable that we came up with called days until 50% of infections had occurred. And if you look at that top bar, the species name is Peltaster fructicola, and compare it to the bottom bar, which is Colatagliopsis FG2, you'll notice that those bars are significantly different from each other. The Colatagliopsis spores were mostly released quite early in the season. That's why the bar is short. Whereas Peltaster fructicola up on the top is a much longer bar. Those spores were released really almost over the entire fruit development period. So one advantage among many of knowing which species is which is we can start characterizing differences in their ecology. And timing of spore release and the phenology of spore release is one of those aspects. And all these insights that we get into the ecology of these fungi can translate into better management. Basically, the take home here is that the timing of infection differs among species of sooty blotch and flyspeck. Nobody knew that before our work because nobody had been able to reliably separate these species. All we had were mycelial types. But now we're getting down to the species level, which is fairly important to do. Well, we had a great many isolates by this time, several thousand isolates of sooty blotch and flyspeck. So we started asking other questions. And one of the questions that we asked was, okay, where did these fungi come from? What was their backstory? What was their evolutionary origin? They have a rather unique niche living on the surface of fruit. How did that happen evolutionarily? Let me walk you through this phylogenetic tree, which looks complicated, but basically the principles are simple. This is based on genetic analysis, phylogenetic analysis of one gene called RPB2, which is commonly used in phylogenetic studies of fungi. Basically, we're using a technique called ancestral state reconstruction to help us sort out where did the sooty blotch species come from. If you'll notice the tree, and these are all species that are extant, they are occurring right now in 2021. You'll notice that sooty blotch and flyspeck species are listed in red. Okay, those are all those red entries are sooty blotch and flyspeck species. The green species here are penetrating plant parasites, that is fungi that their mode of attack is they actually breach the cuticle and breach the epidermis, and they attain the interior of the plant. The third group are the blue fungi, and those are saprobes. They're decay dead matter. So what does this analysis tell us? Well, if you go all the way backwards in this tree, you're going backwards in time. And the guide here is probability, essentially. These programs are all based on probability. And if you go backwards from branch to branch towards the left, you're going backwards in time. And what this analysis formed, and very convincingly, very good statistical proof, was that the ancestors of sooty blotch and flyspeck fungi, which of course live on the surface, were in fact all penetrating plant parasites. That circle, that green circle at the bottom, indicates that when you go far enough back, all the ancestors were high likelihood to be penetrating plant parasites. So somehow there was an evolutionary shift from penetrating plant parasites to living on the surface. There it is. Sooty blotch and flyspeck ancestors were penetrating, that is, host penetrating plant parasites, unlike the sooty blotch and flyspeck fungi that we encounter today. Okay, well, that insight really led to another question, and that is, what happened during this transition? If the ancestors were penetrating plant parasites and the current fungi live on the surface, ectophytes or epiphytes, what events happened at the molecular level during this transition? Well, what was the molecular evolution pattern like? And some wonderful work was done by Dr. Sun Guangyu and his students at Northwest A&F University in China to really penetrate into this area. Let me explain some of the highlights of that work. So first, we need a couple terms. Ectophytes are organisms that live on the plant surface, but they also live in the surface. maybe like in the non-living part of the cuticle and the wax, whereas epiphytes really just live very much on the surface of that surface layer. And it turned out that sooty blotch and flyspeck fungi are really ectophytes. Let me walk you through this. So these are the two first ones are scanning electron micrograph shots. And we're doing a bird's eye view on those top three, looking down at the hyphae and the sclerotium-like bodies formed by the sooty blotch and flyspeck fungi. And that's informative, but we really start learning things when the student, Chao Shu, he popped off some of these colonies and he noticed that they had actually dissolved their way into the wax and partially into the cuticle of the apple fruit. So this was not just living on, it was actually burrowing a little hole into the non-living part, the cuticle on the surface. And you can kind of see a little divot or a little depression here on the side view in this light microscope shot. So the cuticle has been a bit hollowed out by that activity of this particular fungus, Peltaster fructicula. What happened to the enzymes that are used to essentially help a fungus get into a plant, to invade a plant? Well, in the case of sooty blotch and flyspeck fungi, those enzyme systems really were at least partially disappearing. Let me just show you the evidence of Dr. Shu here. This group that I have highlighted with the red bar is the one sooty blotch and flyspeck fungus on this list. The rest of them are classic plant penetrators, Zymoseptoria, Magnaporthe, Colitotricum. Those are your classic penetrating plant pathogens. And here's some biotrophs down here, necrotroph, hemibiotroph. But sooty blotch and flyspeck, when you start looking at the categories of plant invading enzymes, very, very poorly provided with those gene systems anymore. If you look at just this category, which is plant cell wall degrading enzymes, degrading the cellulases, etc., in the cell wall, you can see that Peltaster fructicula, the sooty blotch flyspeck fungi, is rather little presence in these enzyme systems. And the secondary metabolite synthetases, again, another weapon associated with plant invasion, again, very low, and likewise secreted proteins or effectors, very little. So there's, we suspect that there was a strong loss of these enzyme systems and the genes associated with them during the evolution from penetrating plant parasite to surface living ectophyte or endophyte, or ectophyte or epiphyte. But not everything was lost, because if you're living on the surface of a fruit, you need some weapons, you need some defenses. And the circle I have on the upper left is evidence from the same paper that cutinases were very much still a part of the repertoire of sooty blotch and flyspeck fungi. They had to be able to burrow into the cuticle on the surface of the apple, make a little bit of a sort of a foxhole that they could live in. And I think I just lost my. Oh. Do you guys, can you all see my screen still? Okay.
No, we can't see.
All right, let me see if I can get it back.
Okay.
वो भी बता दें लाइंस वाला
Structure.
लाइंस वाला डॉक्टर ना
वो स्क्रीन शेयरिंग की ऑप्शन मैंने बंद कर दी।
अच्छा
ओके
अब ठीक है
It looks fine now.
Oh, it is? You can see it?
No, we can't see again.
How about now?
no it's
Yeah.
okay
It's okay now.
All right. I don't know what happened there, but okay, let's just go on. So the point I was trying to make in with this slide is that Sooty blotched flyspeck did not give up all its weapons when it evolved. These fungi evolved to live on the surface. Cutinase was very important to be able to burrow in. That's the upper left circle. The lower right circle is associated with the activity in this heat map, associated with the activity of melanin. So melanin is really important because it is that dark pigment, gives the dark color to the fungi, but it also blocks ultraviolet radiation. If you're going to live on the surface, you've got to have a lot of melanin in order to survive. Trying to look at the big picture here, if we look down like a bird's eye view on the A diagram there, this diagram, you can see there's the epicuticular wax layer and the hyphae are growing across it, and there's sclerotium-like bodies that are basically just knots of hyphae on that bird's eye view. And then when we look at the side view, this is what's really interesting. You can really see now, here's the live part of the apple fruit, those green cells, the epidermal cells. And look what the sooty blotched and flyspeck fungi have done. They've made these burrows or holes in the cuticle by using cutinase. So they're living on the wax, but they're also being able to burrow in here. So they're not attacking live cells. They're getting their nutrition elsewhere, probably from exudation from the fruit, but they're not attacking or invading live cells. Another good overview from Dr. Xu. This is from his 2016 paper. This really contrasts, I think, in a very clear visual way, what's going on that's different with sooty blotched and flyspeck fungi. So on the left, you have the more common penetrators, the biotrophs, the hemibiotrophs, and the necrotrophs attacking the plant. And once these fungi begin to attack apples and engage or attack the live tissue, there's a whole series of plant defenses that come into play. So you have kind of an arms race biochemically between the host and the pathogen. The host trying to fend off the attack and the attacker trying to overcome the defense of the host. But then you look over on the right, and sooty blotched and flyspeck fungi, to the limits of our knowledge, are not eliciting, they're not activating host defenses, because biochemically the host appears to be unaware of their presence. So all those chemical weapons are not as needed as they are for plant penetrators. Sooty blotched and flyspeck fungi are living there quietly, and that's why we call them stealth fungi, as they seem to escape the notice of the host defenses. I'm coming to the end of my talk here, and I just wanted to mention a few points in summary. One is that this group is really a disease complex rather than two diseases, one called sooty blotch and one called flyspeck. That you'll still read that on the internet. It's wrong. This is a disease complex, not simply two diseases each caused by one fungus. The internet often lags behind reality. And sooty blotched and flyspeck species differ with regard to biogeography and phenology, as I've shown you some evidence of that. I've shown you a little bit of management work where we adapted a sooty blotched and flyspeck warning system for the upper Midwest US. And some evidence that from our lab that sooty blotched and flyspeck fungi evolved from plant parasites. And then we've seen that there are multiple adaptations to the plant surface niche, and that some wonderful work on functional genomics from Sun Guang Yu's lab. One question always in a set of work, we've been working on this group more than 20 years, close to 25 years. What do we not know? Well, we don't know much about spore release. There have not been good studies looking at the timing of spore release. We've looked at trapping spores on fruit, but that's a primitive way to do things. So this is something we have the technology now to do this with using molecular genetics. clarifying the genomics of fruit infection. Well, we have a pretty good idea of how they land and so forth, but we need to know about the events that go on the surface of the fruit. Determining the host range, this is something that's been speculated about. These fungi are mostly supposed to have very large, very wide host ranges because they don't have a close coevolution with their host. But again, very little hard facts. We've only sequenced the genomes of basically 2 or 3 different sooty blotch and fly speck taxa out of 100 or more. This is something we need more depth in. We need to know, I think this is a bigger question now, how similar are these fungi to other surface adapted fungi? We have fungi that live on rocks. We have fungi that live on other kinds of plant surfaces or other kinds of surfaces on Earth. How are these fungi related to those? And I think that's still a somewhat unanswered question. And another area that I think plays into a strength in Pakistani plant pathology, based on what I've seen and read, is looking at the biological control potential for other microbial inhabitants of the fruit surface, such as yeasts. There are many yeasts on the surface of apple fruit, and that's essentially an unexplored area. I want to thank some collaborators here. Dr. Turner Sutton at NC State, basically the godfather of the modern study of sooty blotch and fly speck. Without him, we never would have got started. Sun Guang Yu at Northwest A&F University has done some really, he and his students have done some really wonderful work in the functional genomics of this group, as well as some of the taxonomy work. Gene Botzer has worked with me for many years here at Iowa State, PhD, and then worked for me as an assistant scientist. And this fellow, Tom Harrington, who is a molecular mycologist here at Iowa State, who led us into the mysterious world of molecular genetics. And without him, we never would have dared go into that work. I'd be happy now to take any questions if there is time.
Thank you very much, Dr. Gleason, for such an insightful and informative talk. A huge number of people were listening to your talk. We had a Zoom meeting of 300 capacity and it was completely houseful. So now it is the time for question and answer sessions. I would like to request Dr. Taimur Shakil to please proceed further.
I'm
good.
Okay, thank you very much. Professor Mark Gleason, thank you very much for a very interesting and informative talk. Thank you. I have a few questions from the students. First of all, there is a comment and a question. I will combine it for you. Somebody is asking, he is saying that the fly specks and the sooty mold, how would we differentiate these among the two, while we are having in the Swat region and North Waziristan, we have apple crop. In the rainfall, the disease goes very high. So how would we differentiate and what would you recommend for the control of the disease?
Okay, those are two separate questions, so I'll just take them one at a time. How to differentiate? Well, you know, this is a really good question because you've come on the dilemma that faced mycologists for nearly 200 years. How am I going to separate these when they look rather alike? We have many fungi that look alike. Sometimes we can have one mycelial type that conceals 20 different species. I think the only way to do this is to first attempt to isolate them as we did, and then characterize what the mycelial type is, and then try to do, if you have the capability, try to do PCR on the ITS and large subunit, internal transcribe spacer and large subunit regions of ribosomal DNA. And then you blast them. You take those sequences and you blast them against the online databases for fungi, and you find that where your matches are. There's a nice library of sequences online. It's really hard and discouraging to study these fungi intensely if you don't do the genetics. You know, and I recognize that not everybody's got, you know, a thermocycler, etc. But maybe the possibility is to collaborate with somebody who does have that capability. we're collaborating now with a former student in Malaysia on sooty blotch and fly speck that occurs on mango. That's a project that he is working on currently. But I think it's really important that at least a collaborator have those genetic tools. The other question was regarding management. I wish I had a better answer for you than this, but what we have in the US is fungicides. We don't have genetic resistance. There are no apple varieties known that are genetically resistant to these fungi. Why? Well, maybe one reason is because these fungi don't penetrate. They're on the surface. So the host enzyme systems that would protect against other fungi don't work against groups that don't elicit response from the host. So therefore, we're thrown back on fungicides. Now, that's unfortunate, and to me, it says that we need further research on this group. Maybe we can come up with a biological control. In my book, Pakistan is the leading country in the world right now in biological control. I've read a lot of theses. I've talked to a lot of people. Your country is leading the world in biological control. So if anybody's going to do this properly, it's probably going to be Pakistan. I've seen so much work coming out of Pakistan on biological control.
Yeah. Okay, so thank you very much. We will have one more quick question from our student Mushahid. He said, with reference to geographical distribution of the species, do you think invading a new region or probing a new host might push the fungal species to evolve into a new species? This is with reference to the phylogenetic relationships.
That's a really good question, and I don't know the answer to it. I think some of the dynamics are different here because we don't have host pathogen coevolution in the same way. Some of these fungi may have 100 different hosts. So the host specificity and host pathogen coevolution is probably not such a big deal as it would be in a penetrating group. So they're kind of playing by a different set of rules. What controls speciation, we do not know. In other words, yeah, they have a different playbook. I see Dr. Alam on there. Hello, Dr. Alam. Wakar, good to see you.
Dr. Vakar.
So,
any other questions?
Yeah.
Maybe we can ask anyone from the participants if they want to come ahead and ask any question.
Yeah, okay. I think
Is
we
there
are done.
anyone want to ask any question?
Okay. There is another question. What if we culture both these pathogens on artificial media and on artificial media? So do both of these will spread at the same rate? The question is, if we culture these on artificial media, if the growth rate is the same for both of them?
I'm sorry, could you ask the question one more time? I may have just missed a piece. Maybe there was a little glitch in the electronics. Thank you.
Yes, somebody is asking if we, it is Rifat Ali Khan, she's asking, or he is asking whatever, that if we culture both of these fungi on the artificial media, would you think that the rate of growth on the artificial media would be same for both of the species?
I'll give you a really wishy-washy answer. It depends on the fungi. It's going to be hard to distinguish them based on growth rate. They're very weird in culture. Some of these fungi, when you put them on an agar plate, they will actually develop a yeast phase. They will look like pink yeast, wet and slimy like a yeast. We actually, for years, we threw away plates full of slimy pink yeast. It turned out they were sooty blotch fungi that when you put them on artificial media, they developed a yeast phase. So they're pretty funky. You can't tell a lot from growth rate. I think, I'd strongly recommend, if you're going to go into this group, and there are many unanswered questions, that you partner with people who have access to tools of molecular genetics. It would be extremely helpful and very enlightening, I think.
Yeah, okay. Thank you very much, Professor Gleason. This was a very good interactive session for all of us. The talk was very interesting and we had very good questions and you answered them very wisely. And now we will move to our another guest speaker. He is the president of Pakistan Phytopathological Society, Professor Dr. Tariq Mukhtar. We will request him to have few words with us, then we will close this session.
Thank you, everybody.
Thank you very much.
I am really feel honored and elevated to be the guest of honor in this international webinar organized by jointly by Department of Plant Pathology, the Islamia University, Bahawalpur, and Pakistan Phytopathological Society. We are very happy to listen the valuable and informative talk by Professor Dr. Mark Gleason. I thank you very much, sir. I would take the liberty to say a few words about you. Professor Dr. Mark Gleason, you are very famous and popular among the plant pathologists of Pakistan. Many of our plant pathology students have availed their IRSIP program under the great and kind supervision of you. Professor Dr. Mark Gleason have also evaluated many PhD theses from Pakistan and gave his valuable and positive suggestions. Being president of Pakistan Phytopathological Society, I would like to develop a collaboration with the American Phytopathological Society in future. We will also avail your expertise in future, sir. We are already availing your cooperation. I would also thank all the participants. It is really a very full attended webinar from all the participants from different countries of the world who joined this international webinar and learned a lot from your valuable talk. I hope that the young plant pathologists would have benefited them greatly from this enlightened talk. So finally, I would like to thank Dr. Muhammad Nabi Islam Malghani, Chairman, Department of Plant Pathology, who and his team who took a very good initiative to organize a series of webinars in this time of COVID-19, because we are passing through this dilemma, and I hope we shall soon get rid of this. Dr. Naveed has taken a very good initiative, and this is the second very important webinar organized jointly by Pakistan Phytopathological Society and Department of Plant Pathology, Islamia University, Bahawalpur. I am also very thankful and appreciate the organizers, especially Anna Musa and Taimur Shakil, who did a lot to make this webinar a success. Thank you all. Thank you, Mark Gleason. We are really very thankful to you.
Thank you very much, Professor Dr. Tariq Mukhtar, Professor Dr. Mark Gleason, Engineer Professor Dr. Atar Mehboob, Wardi Vice Chancellor, the Islamic University of Bahawalpur, Dean Faculty of Agriculture and Environment, Professor Dr. Muhammad Iqbal, and the Registrar, the Islamic University of Bahawalpur, Professor Dr. Muazzam Jamid, and the participants from various countries of the world, maybe our teachers and students from various institutions, and the colleagues from various institutions. Thank you very much for your time and for your interest in this series of webinar. the participants who were registered in this webinar, we will shortly, or we will send you the e-certificates in a couple of days. Thank you very much. Have a nice day. Wonderful Ramadan for Muslims ahead. Thank you very much.
Thank you very much, Dr. Naveed, for thanking the participants. And I will, in the end, say thanks to Professor Dr. Atar Mehboob, Vice Chancellor of the Islamic University of Bahawalpur, and the Dean, Faculty of Agriculture and Environment, the Registrar, Professor Dr. Mozam Jameel Saab, and all the participants, and especially the guest speaker. He spared his time, the Professor Gleason. Thank you very much all, and now we will close the meeting and see you in the next webinar, inshallah.
Thank you so much.
Thank you very much. Have a good night.